Okay, good morning, everyone, and welcome again to our regular PAASE webinars. This is the 23rd lecture that we are having for this year. And today we have with us um, Professor Carmen Ablan-Lagman, who joined De La Salle University after she lived for extended periods of time in Australia, Malaysia, Thailand, Japan, and the US as a researcher with a specialization in population genetics and molecular ecology. She established the Practical Genomics Laboratory at DLSU in 2012 through her first grants from the NSF Peer Program and USAID STRIDE program, and has continued on from there. PGL involves researchers and scientists in training who creatively integrate information from DNA and RNA markers with data from image analysis, GIS mapping, carbohydrate and protein profiles, and mobile computing to produce practical solutions to problems in agriculture and aquaculture. Dr. Lagman works with higher education institutions and state universities in the Philippines, academic and research institutions in the region, the US, and Taiwan, NGOs, LGUs, and more recently, the private sector. She received her PhD in marine science from the University of the Philippines in Diliman, and she was a Crawford Fund Australia Master's Thesis Scholar to the Australian Institute of Marine Science, a Japan Fisheries Research Agency PhD dissertation scholar to the National Institute of Aquaculture in Japan, a Fulbright Senior Visiting Scholar to Oregon State University in 2013, and a Leadership for Innovation Fellow of the Newton Agham Program to the Royal Academy of Engineering in the United Kingdom and the Asian Institute of Management in 2018. Dr. Lagman is a recipient of the 2015 Department of DOST Pictured Outstanding Research and Development Award and was second runner-up at the Social Innovation Plus Innovation Competition of the Bank of China, Hong Kong in 2019. Today, she will share with us how to use DNA and RNA markers, image analysis, GIS mapping, LCMS, and mobile computing to address practical problems in Philippine agriculture. So, Menchie, whenever you're ready, please begin.
Thank you. A very generous introduction, Kay, and thank you for the opportunity to sit here in front of you and tell you about the work we do. I know this is a big job, but I'm just going to cut this talk into a few simple applications. Of course, there is more to what we do than what I'm going to tell you, but I just hope to give you a flavor of it. And then after that, I will hopefully give some candid remarks on how you actually we do science in the Philippines, practically for me, who has been home for almost 10 years now, for over 10 years now, starting my research program. And towards the end, hopefully I'll end by 15, 20 minutes. I'm going to give the floor to Dr. Chona Camille Abeledo. She is my first PhD student, finished with my lab just recently and is now on tenure track. Hopefully we'll get her tenure by the end of the year at our department. So may I start? So I put the agencies up there who have actually been funding our research up to this point. Okay, where's my arrow there? So in 2000, I can't remember now, late 2000, early 2000s, Dr. Alcala, if you've seen him here, he was formerly DNR secretary, came up to me and said, Menchie, I have a problem. Can your markers resolve population structure and connectivity among the marine protected areas in the Bohol Sea, a 300-kilometer area? And Dr. Alcala is the father of fisheries management using no-take zones. I thought that was a very interesting question for a PhD because, one, microsatellite markers have just been discovered. The stats and maths and models to try and identify origin of organisms based on profiles of populations was just amazing, and it really caught my interest. The nice thing about it is that somebody was asking for the science. So for like 3 years after that, I would live in this place and work with these people every summer to get my samples and then go back to the lab and do molecular analysis of microsatellites. Thankfully, I was able to get funds to go to Japan to develop my markers there and also Thailand. And after 3 years, this was all going to go away and I now have to go back to reality. But before I left, Dr. Alcala said, please tell the people who you worked with, the fisher folk, the college students in those little islands along the Balsey exactly what you did and why it is important to them. I was shocked. It was not easy to talk about DNA, RNA, microsats, stats, probability statistics, posterior and anterior things to a group like this and still be considered as relevant. And so, with the help of his translating the language, which is the science, and the language, which is the science, we were able to go through that. During coffee, the guy out there, Mang Mario, said, Dr. Lagman, Dr. Kana, so you're going to move on, you're going to get better jobs, better pay, you might not see you again. But please remember when you come back, we will just be here. And that was like something that hit me on the head. Here I was doing science, getting myself up into my own, creating something that I really loved, and somebody that I was supposed to be helping just says, we're just going to be here. And therefore, I left with a sad heart, but I joined an international research institution and I became a research scientist at the World Fish Center, moving around with different regional projects, including something involving the South China Sea. But in 2008, 7, it was time to come home. We had another child, which we didn't have for like 7 years. And so we really decided it was time to come home. But it was hard to get grants, mainly because I was nobody. They didn't know who I was. I did not have a track record in the Philippine institutions. And the Philippine institutions were already full, oversubscribed. Thankfully, NSF Peer had a pocket which was for non-American project leaders, and we had a collaborator, Dr. Kent Carpenter, who actually helped us to bring this thing together. And my friends from UPMSI, UP Diliman University, UP Mindanao, NFRDI, we got ourselves together to create a community for population genetics because that kind of research was in its emergence at that time. And in LaSalle, nobody even know what I was going to do. Thankfully, we were able to get good students, those on the right. And it turned out that all 6 of us were training all 6 of them and some. So instead of Chona just seeing me all the time, she had an opportunity to interact with the others in the team as well. And I thought that was really amazing. We got grant for 330 US dollars. And all of that money had to go through LaSalle. Chona, she understands this. We had to make sure that our partners would deliver. And that would be without the impediments of procurement, of all those stuff that other institutions might have. So doing the science became 2 things, handling the management and doing the science at the same time. This is the NSF peer page. We were one of the first grantees, and there was a lot of back and forth going around between their side of the world and ours. So we were looking into population structure and connectivity of 5 different organisms across the country. And we did come up with the science and the publications and the graduate students that actually got their degrees. I'm proud to say that today, Dr. Chana is on tenure track, and in my department, Ms. Lynn is now in Florida State University, I think finishing in her PhD as well. Apollo is in Norway doing his PhD. And the others, like Ma'am Inkay, is also a professor in one of the universities down south in Mindanao. And they're moving up with their careers. Miss Leslie has just got in contact with us, and she would like to continue with her PhD. What is the element that is of importance there? I think it is an intergenerational community of scientists. Could you see our undergrads at the bottom, our masters and PhDs at the top on the right, the project leaders of different projects, and the people from both sides of the world. What that helped to do is really show each other, where our bars are, like what is it that we jump over in our cultures, in our systems, and it would also seek to let people understand that science has a bar that is common for everybody. So that was our first foray. And when that happened, I now had the chance to start putting projects to the Philippine funding agencies. But before we started to think of what our project will be, we were already stuck with the mangrove crabs because we decided to do that on our first project. So we had to go to the local governments and ask them about the problems of the industry itself. If you see a facade in the local government of a crab that big, you know the crab is important. So we went there and asked them about this, and the problem was really with the juveniles. The whole aquaculture industry, which was the fastest growing, was dependent on these wild caught juveniles. And there were three things that kind of came up: species identification at the juvenile level, molting synchrony, because cannibalism and the soft-shell crab industry would really want to have synchronous molting among all their crabs. And thirdly was temperature and aquaculture site place. Where do they place the aquaculture sites? We don't have the answers to all that, and we are not, we don't claim that our answer is perfect, but at least it gave us things to work on. This is Dr. Chana's first chapter in her PhD thesis, and this is looking into the species identification of juvenile mangrove crabs using the spines on the frontal lobes and using RFLP-based markers. With this cheap marker, you can do a lot of samples and a lot of images, and that was the impetus for now Crabbifier. It's a species identification mobile app which Shauna now leads in her team for the new project for testing and development. So I thought that this is a DNA marker. It is not something that very easy for everybody else to use, but the relevance of it for species identification was made possible by the use of a mobile app. The question on molting was the other thing that was coming around. And this time we were using RNA markers. These are MIH, which is molt inhibiting hormone, and ERK, which is a molt promoting hormone. And to be able to look at both of them, if you see the graph on the side, it is very clear that the ratio of ERK to MIH was separating the group into two. Now we are looking for a morphological character to match this information. And apparently, if you look on the left, you'd see the dactyl, the back sweeping leg of the mangrove crab. It would have a line on the top, which apparently in the literature would say would be changing with molting. And some experts were using this, some locals were doing it as well. And so it would be a good marker if we were able to, for newbies, for those who are not experienced in this yet. So the next question was temperature. One of the things that I got to learn in this process was how to use GIS. And now GIS should actually be a skill that could be put into every scientist, because it is amazing to be able to show your data spatially and temporally. And in this case, on the same island, we were just looking at temperature, a very simple algorithm to determine which one is hotter most times of the year than the others. And just by coming up with a graph like this, you can tell people where the hotter areas will remain hotter longer and the cooler areas will stay cooler longer in the decision making for siting. But with long-term data, 5-year moving averages, we found 3 areas, Bataan, Bicol, and Cagayan, with differences in temperatures that were appreciable, and that they were also in different climate regions of the place. The concern here is with the increase in aquaculture of mangrove crabs, the supply of juveniles will be limited, and there will be transfers. There will be movement. Is it worthwhile to transfer something? I know we were touching into the area of adaptation, the big A, which ecology is going to be having difficulty to work with. But we now had a new tool, and it's called transcriptomes. Transcriptomes are able to record the whole of the expression in mRNAs of an organism under different conditions. And so we started doing that, Bataan, Cagayan, and Bicol. Bataan is on the west side of the Philippines. Cagayan and Bicol are in the right side. Historically, the east side and the west side of the Philippine marine systems are known to have a divide from long-term processes. So we were expecting Cagayan and Bicol to be similar to Bataan. And indeed, it was true. If you look at the two images on the left and the right, these are Cagayan and Bicol. And the marked black and red dots would be your upregulated and downregulated gene. Bataan was different. However, if you look at the pattern of upregulated and downregulated genes, Bataan and Bicol had more upregulated genes than downregulated in terms of trend. So it brought to us a very interesting problem or question. Is this genetic historical? Is this exposure? And we are working on this, and we will be submitting the paper soon. So even with the heat maps, you could see the differences in the between and within of the populations. The interesting thing though is before we can do all that, we had to also, not had to, we wanted to. We wanted to help the local communities in the areas that they would want to, to learn. And so even before modules were in vogue, we were coming up with our own. Oops, you can't see. This is our modules. We have come up with modules for GIS, and here's another one. It's called DNA barcoding. So yeah, with the number of requests and people thought that what we were doing was interesting, we had to come up with at least exposure seminars for them. And that's how we did science. That included molecular work as well. You will be amazed in how much infrastructure exists in the state universities and colleges for molecular work. You see, when we went here in Palawan, this is a state university in Palawan, their equipment was even better and more advanced than what we had. But they did not have consumables, they did not have water, and they did not have people consistently able to do this. So we did something initially, which was to give them exposure on the potential of all of these, both theoretically and practically. We brought all our chemicals with us. And that was an interesting thing to do. So in summary, for that part, we have modules, we have training, we are looking for practical applications from the DNA, the gene expression, and the transcriptome analysis part. Sorry, it's not moving. So what I soon realized after this was that this is not sustainable. It's very tiring and you're giving so much and it is not sustainable. And it was for this reason, we started to move on into another direction as well. But we continue with the other. We have 2 other projects there. And this was with Dr. Mojica in coffee. Dr. Mojica and I were in the same batch in a Fulbright Senior Visiting Fellowship to the US. And we happened to meet each other at the Lyndon B. Johnson Center in Texas. We were encouraged to go for a seminar that was totally different from what we really did. And I went to social entrepreneurship, and he did too. When I asked him what he was going to do when he goes back, he says he's going to be the director of the National Coffee Resources Research and Development Center. And when I asked him what the problems in coffee were, he was talking about variety identification. He was talking about the presence of seedling, particularly for a group called Liberica, which is what we know as Barako. I got me and started me planting my own trees as well. And this is what you see on the left. I had a small ledge, and now my tiny trees are about 9 months old, ready to be distributed to some friends. They have gotten in from STC, it's LaSalle. The other thing that also got us into the interest here was to develop DIY, farmer-friendly tissue culture methods for coffee. We were able to somehow come up with, there is a lot of literature out there that doesn't need big high-tech equipment to be able to work on this tissue culture kits. And we started to do that in the lab. And we have had success. We have had failures, but we do have success. This way of doing science was something that caught our attention. And the sad thing I find is that we are very time bound in terms of manpower. If you lose one person who's working on something that is of very interest, you're going to be stuck and you're going to go down from scratch again. And then you have to keep the next person up to speed. So when we started wanting to work with industry, that was my biggest setback. If we want to be sustainable and scalable, it's inevitable that you have to work with industry. And so we are having, now that COVID pandemic has come around, it's basically things in the laboratory have stopped. But we have done most of the other work that we can do with our partner institutions. And that's why partnership was key. We're working with CIFDEC in Binangonan, and we also have working with the chemistry group of De La Salle University. Finally, a breakthrough came when we started working on the mangrove crabs again, and I met a few of the industry people that were working on these crabs, and they basically said they are worried now more about disease. And who is not, given the reason we're sitting at home is because of a potential disease. Mangrove crab aquaculture is the fastest growing yellow line of all the four organisms that we actually cultivate. The fifth one was seaweed. It is interesting that the low survival of the disease from disease of the juvenile is also piling up aside from the other things that we already know based on what we've discussed with the farmers and the local governments. However, if you look into the literature, there's over 26 identified potential sources of immunostimulants for feeds, tested, published. What are we going to do? Do we mount up feeding trials for each of these 26? And if you look at the parameters they use to evaluate each of these, they are different biochemical markers, different threshold levels, different treatments. So we proposed a project that we will actually look at, let's say, 3 or 4 of these, and starts intensive experiments on using a functional approach of transcriptome single gene responses of the crab, analysis of the sources as well, and bring all of that together, bring all of that data together, hopefully be able to come up with a story with markers that we should look out for in terms of the potential as an immunostimulant, and the next one was the response of the crab. So if we have these markers, then it would be much easier to go over what we know is already potential, so that when we come up with a result at the end, we are standing on somebody else's shoulders and somebody else's investment on this potential feed additive sources. And hopefully the objective is to come up with a novel feed, which is field tested for at least this first batch of the year, and then a set of diagnostic markers, whichever way that would be, that is jointly validated from different sources of data, to be able to say that of this 26, we work with 1, 2, 3, or 4, and then work on other material that is possible. We don't have to do all these things with the same materials. We have a set of markers for farm wastes that is also possible for fruit peels and all that sort of stuff. So yeah, it has been a very long journey of wanting to be relevant. It has been a very long journey of talking with so many people. And to be able to do science means investing so much in what Mang Mario wanted, which was not for himself, but either his kids or at least for his community. So I really thank everybody, because I have had a very amazing team so far. Dr. Michael Puruganan is one of the guys that we've working in the coffee, Dr. Weng Eguia, now Dr. Anish Shrestha, and Dr. J.I. Hanairo. Plus, Dr. Chona is now running her own project, and everybody else in this team has been amazing. So for the next minutes, I'll just pass on to Dr. Chona so that she can also give you a perspective of how it was like going through the past 10 years, because she and I basically came to LaSalle almost the same time.
Thank you so much, Dr. Menchie, for this opportunity. And also, I guess thank you so much for the past 10 years also. This has been a very amazing trip down memory lane for me. It's been 10 years. Oh my God.
Yeah, much older.
So, I think one of some of the key aspects that I've learned in the past 10 years while working within the practical genomics laboratory of Dr. Lagman is about how the mentorship system in the Philippines, but I don't know if it's really the mentorship system of the Philippines or just a unique kind of mentorship that I had with her. Looking through her presentation or watching her presentation, I just realized how much of our work then has really influenced the direction of the research that I want to go into now. So just to give everyone a background, the new project that Dr. Menchie has mentioned that I've been working on is called Crab Tech. And Crab Tech is deploying some of the technologies we made during the first stages of our mangrove crab project and distributing it into different fishing communities in the Philippines. Of course, we're set to go to Visayas this year, but due to COVID-19, some of our travel plans have been delayed. But because also of the magic of technology, we're trying to work with different Bureau of Fisheries units to disseminate our workshop materials. But what I've realized is the way we do science is a little different. Whenever we do our talks and we do our lectures, I have two things that surprise me always. The first is that um, we only have a limited amount of time that we have, and majority of the scientists at my age are running after publications. And please don't get me wrong, I'm running after publications too. It's just that when you're running after fishermen in the process, sometimes the publication tends to get a backseat. And I notice why, mostly because I guess I've been raised in an environment where the people who will adopt the technology should be at the forefront of our minds. So, in Crab Tech, one of the first things that we've done is transform our materials into different languages. So I actually have different booklets here at home that have descriptions of how to use our technologies in Tagalog, Bisaya, Ilocano, Waray, and all of those things. And Doc Menchie would attest to this during her first few weeks because I know she speaks Ilocano and a lot of other languages. Ma'am, tama po ba? Is what we're saying right? Is this the way to say it? And a lot of the other scientists in the lab also speak different languages. So they would suddenly put in a chorus of different ways to say something. And it's it's been it's been an exciting but also challenging road. And while we go into the different places that we've been, at least in Luzon, we've seen how our technology can be useful in some aspects and how it's not useful in other aspects. So, as scientists, we have a particular perspective about the world, but meeting the people on the ground really changes things, I guess. So, that's one of the main things I've learned. But I think one of the reasons why we were able to maximize my being a PhD student in Dr. Lagman's lab is one of the first things we did is to clarify both of our expectations with each other. I remember back then, I I think it's every Filipino scientist's dream to study abroad. But at that time, I was barely financially stable. In fact, my work, which is teaching in DLSU, couldn't become stable if I don't get that PhD degree. But at the same time, I couldn't really just fly out because my family needed me here because my mom was getting old. I'm a menopausal baby, so my mom was getting sickly at that time. So I was planning to delay my PhD later, but Doc Menchie gave me this amazing offer at the perfect time. And she had this very clarifying set of words to me that I believe her mentor also said to her before. Sometimes beggars can't be choosers. I wasn't in the best position to choose what my future will be, but it is up to me if I wanted to be in a position of power over my situation or in a position of being just a victim forever. So I decided to take her up on her offer. And yeah, I guess that started a roller coaster ride of amazing things for us. And another thing that I think I might have misunderstood, but I don't know, was that she told me that, oh, this is an amazing opportunity because this project, that USAID project, will fund your dissertation. And that was amazing for me because, like I said, I don't have the money to fund my own research. And in my head, I understood that as I only had 3 years to finish my PhD. So in my head it was I have to respect my mentor's time. She made sure I have funding, so I have to work. And so I did. certain times, yes, of course, it was tiring, but I also had an amazing support system. My family supported my dreams. My boyfriend, now husband, supported my dreams. So everybody around me respected what we wanted to do. And I guess one of the reasons for that is they can see how our research can potentially benefit people. And I guess one factor for that is how we document it. We like to talk about our research a lot. And that that also taught us how to talk about it to people from different disciplines. So I really learned an appreciation for science communication then. And I guess until now that really affects how I do science communication now. But the best support system I had then was the fact that I respect my mentor, but I also wasn't afraid of her. Every time I messed something up, she was the first person I talked to. And I think that's a very important thing to establish with both our students or our supervisors is that you shouldn't be afraid of making mistakes. Because as a student back then, I think that would be the worst thing to do is for me to make a mistake and not have anyone correct me. And Doc Menchie has created this environment then wherein if you make a mistake, sure you owe up to the consequences, but she will help you get through it. She won't abandon you, she won't scream at you. It's not that. So I was never afraid of that. I just whenever I would feel scared and whenever I would feel worried, she is always the first person I talk to. Because I believe that's the best kind of mentorship relationship where you're not afraid to mess up. Because that's our, I mean, now as a project leader, always afraid to mess up. And yes, Doc Menchie is still there, but she also has her new kids, right, to worry about. And I have to have the courage now to stand up on my own and mentor too. So I have to open those doors for my own kids now. But yeah, I mean, as a student, it's such a unique opportunity that for a short period in your life, you're allowed to mess up and someone will catch you. Someone will help you. So yeah, and I guess at the end of it, since I've went through an amazing mentorship for the past 10 years, the next steps would be to reciprocate and to give back. So I try to incorporate as much of what I learned from her in this new lab that I'm trying to build in future projects that hopefully someday can actually happen after COVID-19. I'm still trying to adjust what research would be in a non-laboratory setup since I've been in the lab since I'm
All our
supposed to
lives.
be. Yeah.
All
So
our lives.
yeah, so it's a strange, brave new world. But yes, so thank you so much for this opportunity to share my experience. And yeah, I hope it has added some value to this morning session. So thank you.
Thank you so much for that, Menchi and Shona. So I'd like to invite Alice, will be moderating the Q&A.
Hi, good morning.
Yes.
So thank you very much for that presentation and at the same time for sharing your research journey, answering, of course, research questions. And we saw how you were able to grow your lab and have that culture, research culture, mentoring the students. So we have our first question posted in YouTube. This is from Raymond Tan. So he mentioned, could you share some more of your insights on bridging the divide between scientists and counterparts in the industry? What's the biggest barrier and what might be the key to overcoming
Those
this?
barriers. Yeah. The realization that we have to work with them came when you get so tired already, always trying to find money. You suddenly realize that it will not be sustainable. And even if you have a solution, it will not be scalable. Because other people, you have a million, for example, and your technology costs you 100 pesos. So 100 pesos over a million, 100,000. After you're out, you're out. So your technology only gets that far. But if you ask somebody to pay for it, even if it's 100, it can go forever, right? So it goes back and then go back and go back. And that was the reason I tried to look for an opportunity to train in this area. And I competed for things like LIF. I wanted to understand this more. Now, sitting in those things, one thing I realized is the greatest barrier for a scientist talking to industry was to be where they are. They sit in a different place, we sit in a different place. They talk a different language, we talk a different language. And so trying to bridge that divide was hard. And thankfully, Grow Asia came along. For some reason, Grow Asia was able to come up to us in LaSalle, came to our lab, and their objective was really to have small-scale farmers. Remember, my heart is with small-scale farmers, small-scale fishers. And if your heart is there, how does industry come in, right? How are you, if you want that at that level, you will be more giving rather than taking and making things roll. So that's the first thing. Scientists, you have to break that part, because we grew up too much on grant making and subsidized operations and not thinking of ROI at the end of the day. Because it's research, right? It's research. Efficiency, projects should not be running 3 years. They should be 1 year, something out, something like that. Because if it was a private company running with you, that is how they will think. The second thing was, listen. So in those Grow Asian meetings, I went to where two feed company people, I research their names, I know their faces, I go to the meeting, they don't know me, I know them. I know who they are and I know where they're sitting and all that stuff. So I went to where they were going and I listened to what they needed, what they were talking about. And that's when I realized, wow, we are triple, quadruple whammy in this disease thing. It's everywhere. And then I was also in the scientific circles. Remember, Doc Chana, we sit in the discussions of other projects of the OST. We sit in those reviews. And we see, at least for me, I see one by one perspectives. There's this new something, it can help, let's make it into a feed. That's new something, it can help, let's make it into a feed. And that's when you started to read the literature. That's the next thing. Don't read the literature when the problem comes up. Just be kind of broad-based in the thing. And present yourself. Talk. It's hard. Doc Shana, I remember, it's hard. Because we have a sort of, not really inferiority complex. It's more, it's just that we're different. This is where your SUC counterparts will be very, very useful. Our friends in the state universities and colleges, our friends on the ground, they understand this so much more than we do. And those long talks over coffee with Dr. Mojica were like just amazing. So we understand somehow, and then we put up and bravely talk about these things. So now when we're doing this feed thing, my idea was we get the money, we've set up within a year. By year 2, we should be on the ground testing the feed. We don't end the project with just the product. It should be a product that has been tested. And so from the start, when it was an idea, we were already talking to people so that they will raise their hand and say, okay, do it here. Do it here. And then you kind of ask how much is it gonna cost? And that's when I think Dr. Al Serafica is here. That's when I give you a call, Al, right? That's when I go, Al, what am I gonna do? Don't know what I'm gonna do. Those are the times that you're treading on not so sure ground. But that's what Dr. Chana was saying. If you know that when you're gonna mess up, somehow you're gonna mess up, something's not gonna really fall flat on your face. You're not afraid to just run in. And there have been a few run-ins, and I would just tell people above me, Dr. Raymond, Dr. Al, there's this thing, there's this opportunity, what do you want to do with it? That sort of thing. But, it's really way out of your teaching, it's way out of your academic requirements, it's way out of your, I'm graduate school coordinator, I'm teaching 10 units, and I have 2 projects with 11 people in the lab. So it's a passion, but at the same time, you just have to get out there. You just have to make the time for it, I guess. And don't promise too much.
Yeah, I think Al has something to say.
Yeah.
Yeah, I mean, definitely Menchie has been my classmate in batch 83 in Philippine Science. I've been wanting to
So
really
weird.
get a few spin out companies from her, and I think it's about time. I mean, we've had a good run with grants and partners, but I think really forming teams around each of your technology, Chana and Menchie, to be able to pass it on to them so that you open up some other opportunities for you. You're good communicators, but again, the business aspect and sustainability needs to be handed off to somebody who lives and dies on the business as well. The same way you live and die for the science. So I think we just need to again form our teams moving forward, and definitely while we still have Raymond and Aris and Alvin and Kate and Lasal, I think we'll be able to eventually get this out. And hopefully, I've used Lasal Laguna definitely for the spin-off companies with Noni Santos as well. But I think this is the time. I'm feeling that you have enough field data available for you to be able to make a convincing part. I think it's industry and potential partners, both funding partners to match your existing efforts already from the science part is the key. I think you're almost there. You're on a tipping point already. I view it
No
in my
COVID.
time. Yeah, yeah. In my time in my microbial bacterial cellulose development, you're almost on the cusp of a venture capital funding situation. You're 5 years in, so I think
Yeah, we're 5 years in. Yeah.
probably yeah. So I think we're probably ready for that. I mean, it takes longer here in the Philippines because of both very risk averse capitalists as well. But I think you have enough material for them to be able to make that leap of faith because of the risk that you yourselves have taken and already done some proof of concept. Thereby, field testing is already there. It's a matter of now scaling up for mass dissemination. So you're right there. You're on TRL5. So we will work on this definitely. Yeah. Thank you very much for sharing both your experiences, and of course, I have 10 technologies out of LaSalle that we need to get out there spin-off company before I leave you guys. So let's do it now.
Oh.
Thank you. Good
Thank
job.
you, Al, for the comment. We
Good.
have a couple more questions in our chat box. First, by Asuncion Raimundo. Hi, Dr. Lagman. Is your novel feed product already being used by the aquaculturists? How do you convince them to use your products based on RNA and protein marker results?
We are in lab testing, ma'am. We have a partnership. That is the key. If we involve them at the end when we already have the product, they will just look at us and say, "What is that?" But if it starts with them involved also from the time we were starting, then they will say, "Oh, how is it? How are you going? Are you ready?" So it's two different kinds of responses. We have been working, we have a chance to, I believe that we are also given opportunities. Many of the things that happened is out of my, something just comes out in the email or somebody calls you on the phone. And this one was with Rojas City. And Rojas City is the biggest aquaculture capital in the Philippines. So the people there own 700 hectares at the time of ponds. And I go like, wow. So yeah, now they're just waiting. So we have to speed up what we do in the lab. That's the difficult part when you start working with these people. They want your product and they want it now. And that would mean we cannot slack in the lab. We cannot slack as a graduate student will. And maybe that's why some people get angry, but what else can we do? We're a teaching institution. We teach. I believe a lot that more is caught than taught. So be very careful how I actually do stuff. But anyway, so yeah, ma'am, we are there. We've done 2 runs already. We have enough data. Our next step is going to be field testing. And we've had 2 things set up, one in Rojas, which is very far, and another most probably in Palawan or Giwan. They're the ones who told us, please come. It's not like we're trying to push something. They're the ones saying come.
Thank you, Doc Menchie. Another question posted by Tadeus Carvajal. Dr. Lagman, what are the setbacks or challenges you experienced when you were trying to set up your laboratory unit in the DLSU? What advice can you give to junior researchers who are currently setting their own laboratory?
Chona, you start, and then I go.
Okay. Hi, Sir Tads. Thank you for that very challenging question. Yeah. I think one a major challenge is always trying to understand how a research lab works and how um, a bigger system, a bigger organization system works, and find mechanisms to make them complement each other instead of going against each other. Because I think at the start, you have your deadlines, and then of course accounting, procurement, they have their deadlines and their processes. And if we don't take the time to actually open up the conversation and figure out how they work and how we work, it's always just going to be a frustrating mishmash of processes. And that's the most frustrating thing, when as a laboratory we end up being more focused about processes than the actual research that we want to come out with. So one of the good things that Doc Menchie made is that, okay, when we open up the next project, we will bring accounting and procurement and everybody in a meeting so that we all talk about what we're going to do. So at the onset of this newer project that we have, we made sure that when DOST was orienting us, people from the different offices in DLSU were also there so that they know what we're up against. And every time we come into an issue, we remind them, don't you remember? And things ended up becoming smoother. But yeah, that's very challenging, figuring out how it is for other people, because our job is difficult enough as it is. But when you see that you have to collaborate or coordinate with others, it makes it harder. But at the same time, you can't get anywhere without coordination and collaboration. So yeah.
For me, I made a few mistakes. I must say, I was a new gung-ho researcher, as everybody else, I guess. And wherever you go, the first thing you have to know is, where is your perimeter? Where are your boundaries? When do you know that you're crossing them already? And at the start, it was hard because I was not from the system. I was from somewhere else, right? So it seemed like I was like pushing walls and then see if there's a wall there. I'm pushing the boundaries to see where the wall is there. And it came to the point that I really wrote the boundaries, and please tell me, are these the boundaries? That sort of things. Particularly if you have partners, 5 partners, what if they do something? It's my neck that will roll each time. 1, find your boundaries, find your champions, find hopefully somebody who's going to get your back and get, that's why I always don't move and justify, ask before you move. That's what I always tell my team. Don't just do something and then justify to me why. If you're in doubt, you ask first. If you're doubt, don't. But that should not stifle creativity. Create an environment where that's what Chan is saying. Allow them to make mistakes so that they can ask the question. Second, I have 3 things in the lab that I've always underlined, that Chana, right? Cross this line and then we're in trouble. We're through. Integrity, humility, accountability. If you're not coming to work, tell somebody. I'm not going to say don't not come to work. You can. We're on flexi time here. Because if you're holding millions and we don't have integrity, do you want me to check every single centavo that gets out? I cannot. So if I cannot trust you, we're through. If you're not accountable, we're through. If you're not humble, even if a smallest person in the institution drops a paper that you need badly, you can be in trouble, right? So that 3 things, and Sean has seen that as well. The lab has seen that. There are people that we have had to let go simply because the lines were crossed. So I'm very specific about lines. That's what I was from the start, because I did a lot of mistakes. People will be attesting to that. Simply because I wanted to push the line so far in time. I hope that makes sense.
Yeah.
I hope that makes sense.
So I guess that would be the questions posted in both YouTube and here. But I would just like to pose a question, or perhaps, Doc Menchie, would you have any words for our young and, of course, starting researchers who would like to, of course, uh, progress with this research career?
It should be a passion. It should be a mission because it's not easy. And when circumstances get difficult, you should know where your priorities are. Syempre, when you're gonna cross my family, sorry, I'm gonna drop you. Clearly, that's not a question you ask me, right? But the other thing is collaboration. Much of the double efforts or even the things that are impossible are made possible simply because we're able to collaborate. Can you imagine working on crabs in our setup in that time, Doc Shona, in 2008 or 7? That would mean we will build recirculating systems. That will mean we will hire people to do this and that. That will mean we have, so many other things. But through collaboration, we are still able to do what we do, even in COVID-19. Even in COVID, we can outsource what we need to be done. Dr. Drexel, Dr. J.I. have been very amazing in this. Dr. Weng, Dr. Michael Puruganan, all these collaborative things. These things we have to create, and it's scary because you have to trust. Imagine holding, magkano na tayo, Doc Shana? Anyway, if you're holding your millions and you're trusting somebody else to deliver and trusting somebody else to value it the way you do value it, and if something messes up, it's your neck that will roll because I'm the full-time faculty in LaSalle holding all that and signing all those. It's really scary. So I was telling Dr. Chana, I didn't realize it's going to get this far. But we have to, rethink after COVID. We'll think about it. But for young researchers, interdisciplinary. The fields are so, chemistry is doing DNA, engineering is doing microbiome. It's amazing. I really think it's just an amazing opportunity to be able to do all of these things together because your world is now less siloed, more integrated, and to be able to negotiate that, I think will be your competitive advantage, which I would say was mine. To be able to break silos and create collaboration was something I seem to be only be the one to be able to do. I'm not making yabang, but initially, and that became a competitive advantage. So find your competitive advantage. But do your science good. Publications are non-negotiable. Graduate degrees are non-negotiable. I hire people who are students. So you kind of figure out how you're going to do your project and your studentness at the same time. And their research in their PhD and MS is not the project proposal because that's mine. I will fund, we will work, but this is your job, that is your research. And then look only at me. Don't look left, don't look right. Look straight. And that's how I was telling them all the time. Look straight. Because it's not that, I'm talking too much, but I'm saying build your vision with your team. Tell them where the boundaries are so that there are no questions at the end, and that be willing to trust, but also be careful.
Thank you, Doc Menchie. We have another question posted by Mary Michelle Ginto. Is there such as too late to know this type of technology, especially if you are just about to learn it for your PhD?
Kind of not sure if I understand. Is your technology late? Basically. I don't think so. It's always a competitive advantage. Not just because you did another kind. This is what I'm saying for my field, population genetics. We started with allozymes, and then we moved to microsats, and then we moved to single gene sequencing, and then we moved to concatenating genes, and now we're doing whole genome. if you're doing it, what does that mean? What you do now is correct and what was done in the past is wrong? Or if you don't have the technology of whole genome, then it's not worth doing at all? No, I don't really think that's it. It's a matter of crafting. As I was telling Dr. Bella, now we're doing SNPs and then we're doing, what do you call this? Cutting RFLP for the SNPs. It's a backward step. Dr. Bella was telling me, isn't that going backwards? And then I said, we are going forwards, backwards. Because with the backwards of cutting SNPs, Chona can do 1,000 samples with 20,000 pesos. But with sequencing, she can only do 500. 200. The data is required for an app. What do you need? You need the RFLP. You don't need the sequencing. So not just because you're not using sequencing means it's wrong for that particular application. So yeah, it's really, I was going to tell, what do you have? Deal with it. Come up with a track record. Show that you can do science. And I'm sure people who review are not really dependent on which technology you use. It's more the novelty of your contribution.
Thank you, Doc Menchie. I guess that would be all the questions posted both in YouTube and in our chat box. So again, thank you for an insightful presentation. Before we actually end our session, let me just share my screen. Just to announce next week's presentation. So it's entitled Monitoring Fish Biodiversity Using Artificial Intelligence. So this would be presented by Professor Prospero Naval Jr. So Dr. Prospero Naval Jr. is a professor at the Department of Computer Science of the University of the Philippines, Diliman, and laboratory head of the Computer Vision and Machine Intelligence Group at the same department. So that would be next week, same time. And the registration link is also shown here with you. So I guess that would be it. Dr. K?
Yes, thank you so much. Thank you for our audience. Oo, kasi before 9 pa naman pala. Yeah. So thank you so much, Menchie, Chona, Aris. Si Aris talaga. Thank you so much for this morning. And I hope that we see everyone again next week. So I think it's a bit, it's a bit related. At least we started talking about agriculture, fisheries. So next week we'll have Pros naman to talk about how we can apply artificial intelligence for our fisheries. So thank you guys. Have a good day ahead. Good night or good evening for those of you in the US. Thank you.
Thank you.
Bye.